Today we're going to complete our lecture 10 with part B, and we're going to look at the concept of a variety of things within this model of our action potential. And within this, we have the idea of threshold stimulus. And the threshold is the point, usually in a lot of these models, at negative 55 millivolts. And if we're at a resting membrane potential, say of negative 70 millivolts inside the cell, if we don't reach our threshold of negative 55, we don't have an action potential. And so threshold stimulus is the weakest stimulus to get an action potential. And once we've reached the threshold, then we have the all or none principle, which says that once we have that threshold stimulus, we're going to have an action potential conducted along the entire axon at a constant and maximum strength. So we fire these action potentials at each of these nodes of Ron-VA, and once we've started those at the axon hillock, then they fire at each node of Ron-VA, and we see that exact same action potential graph in terms of millivolts representing sodium in and potassium out, and then our restabilization of resting membrane potential. If we have an impulse or a desire to have a movement, but it's not quite enough to create some kind of, say, muscle movement, then we won't quite fire off enough neurons to actually achieve that. Yet if we are able to reach threshold, then we can have action potentials at each of those nodes of Ron-VA, or this could also represent different nodes along a sarcolemma, and we can fire those in as much of a quantity as we need. This concept of how many action potentials we fire is the concept of intensity, and within that we take a look at frequency modulation. If we have an increased threshold stimulus, then we have increased firing frequency. The more times that we hit threshold, the more times we're going to fire a complete action potential. Recruitment then is the idea that if we have increased stimulus intensity, then we're going to have increased axons within the nerve firing. The more stimulus we have, the more axons that we're going to fire. When we look at our chart then of action potential, we're at resting membrane potential, say at negative 70 millivolts. We hit threshold at negative 55 millivolts. We have sodium come into the cell membrane, very positive, moving us up to usually like, say, a positive 30 millivolts. Then these sodium gates close and potassium channels open, and we have potassium leave the cell. Potassium leaves the cell to such an extent that we hyperpolarize, say, to negative 90 millivolts, and then we utilize a sodium-potassium ATPase pump to go ahead and reset our resting membrane potential of negative 70 millivolts. So within this, we have an absolute refractory period and a relative refractory period. The absolute refractory period is this point where sodium is coming in and the sodium channels are slowly closing. Then our relative refractory period, where we could still fire an action potential potentially, is when our potassium is leaving the cell. But we can't fire a action potential during the absolute refractory period because we're not reset or adjusted enough to go ahead and have that action potential be successful. We've looked at this model of the nodes of Ron-VA. At each of these nodes then, when we have a change in polarity, we continue the current signal down the axon. While we're having our reset, we have a refractory period that allows for a close off of a potential for action potential, and it also makes it so that our signals can't just go back up the axon, since we're closed off to having any action potential for a period of time while the sodium gates are closing and sodium is coming into the cell. So the absolute refractory period, we have sodium coming into the cell, and then we also have our sodium gates slowly closing as our potassium gates open, and we have that absolute refractory period while we have this period here, which is a relative refractory period, as we have potassium leaving the cell, and we're not completely comfortable to fire an action potential until we're back to our resting membrane potential. but it is possible to fire an action potential within the relative refractory period. Absolute refractory period, no action potential. Relative refractory period, if we need to, we can fire action potential. Synapses then are these connection points where we have communication between neurons. We have axosomatic synapses. Axo with the axon to the soma, the body. We're coming from some other axon into the body of a neuron. Then we have these axoaxonic, which would just say we're coming from one axon and going directly into another axon. And then we have axodendritic, where we have axons coming in and communicating with dendrites. Chemical synapse is one that we've been looking at within our muscle unit, where we have these ligand-gated channels that receive a neurotransmitter or a hormone. And what we've looked at in particular is acetylcholine. Acetylcholine being the main chemical that we've looked at for these chemical-gated or ligand-gated synapses. A lot of just images based on that. These being vesicles coming up to this end plate area here, ready to release their neurotransmitter. When we look at the situation after a synapse, post-synaptic potentials, we're interested in this area where after the synapse, we have released some neurotransmitter, but the question is, did we release enough to go ahead and have an action potential? If we have an EPSP or an excitatory postsynaptic potential, then we're going to excite the cell to go ahead and change its polarity enough to fire an action potential. If we don't reach threshold, though, we don't have an action potential. If we fire an IPSP, an inhibitory postsynaptic potential, then we actually can hyperpolarize and create a more negative environment so that we definitely don't fire an action potential. And in our construction within human existence, we're more primed to be performing more inhibition on a regular level than excitation. Single channel allowing two-way traffic would be this idea that if we have these channels that allow for inflow and outflow of different ions, then if we have enough of a change that is excitatory enough, or excitatory postsynaptic potential, if we have enough of that change, we may fire an action potential if we reach threshold. So just this idea that we're in this negative internal cellular environment with a lot of positive charge in our extracellular environment. If we can change this intracellular environment to be positive enough, we can begin a cascade of that depolarization to then go ahead and have an action potential. A single channel that allows another type is one where we might see chlorine, for instance, it's more negative. It is going to have an inhibiting effect on the cell. We could hyperpolarize and reduce the likelihood that we're going to have an action potential at the axon hillock, this triangular region where we usually fire off action potentials. The voltage-gated sodium channel then, just that idea that if we're closed, then we're not allowing sodium in. If we're open, we are. And that is going to allow for an increased positive environment within the neuron to potentially fire off an action potential. Our voltage-gated potassium channel works similarly, except we're going to have potassium leave its saturated environment inside the cell and flow to a lower concentration environment outside of the cell. And these are, again, voltage-gated rather than chemically gated. So they're opening and closing based on changes in voltage. And just this idea, again, that we see reiterated that we fire these action potentials at every one of these nodes of Ranvier, for instance, until we reach the end. So the postsynaptic response is the sum of all inputs arriving at the synapse. A single excitatory postsynaptic potential is often not enough to trigger action potential in a postsynaptic neuron. We usually have neurons working in concert. It's not this idea that one neuron is actually going to create the entire change. We're going to recruit a lot of neurons to do that. The recruitment idea then is summation. If we have one input, an excitatory input into a neuron body, so this could be an example of an axosomatic, we're coming from an axon directly into the soma of the neuron, that single input may not be enough to fire our action potential. If we go ahead, though, and fire in close enough succession, we release a little bit more neurotransmitter, we can actually then bump that to have the action potential. If we go ahead and recruit another axon, then we have 1 and 2. We can have spatial summation, which would say the combination of these two axosomatic inputs could give us an action potential. And then we also could have excitation and inhibition happening at the same time, where we inhibit, we hyperpolarize, and then we excite, but it's not enough to reach threshold. So just a couple of different looks at how we can have a summation of these inputs into a neuron to either fire an action potential or not. Looking more closely then at temporal summation within that model, this would be many EPSPs and or IPSPs if we're inhibiting. Those can come in temporal summation in rapid succession from a single source. The important thing to notice within these different ideas of summation is where is the input coming from. In temporal summation, temporal where we have multiple inputs, we've seen that before, it's going to be these multiple inputs from a single source to get us to an action potential. And in this case, we look at excitatory, but this can also happen in an inhibitory postsynaptic potential as well. Spatial summation then, we're looking at more than one axon involved, many synapses over a broad area simultaneously to fire that action potential. We also have this idea of neuronal pools. Neuronal pools are many neurons organized into functional pathways, and most activity of our nervous system is not a function of a single neuron. It's going to be a function of a neuronal pool. The arrangement of neuronal pools then is going to determine activity. The types of activity that we have would be amplification, divergence, intensity, and duration. We need to take a look at those. If we have diverging circuits, we have this idea where we could have amplification as a diverging circuit, where we have this start-off input that then recruits more and more neurons to amplify that original message. And through that amplification, then we're going to have this output that is going to be divergence in the same pathway, amplifying it. Divergence inside of the grouping of a diverging circuit, still the same idea of one input, but as we hand that off to the successive neurons that we're adding into the mix, we go ahead and have this signal split to do two different things. So these can be both sensory and motor. They both have an amplifying effect. In both of these, we're starting with a singular element and we're ending up with a pretty large recruitment of more neurons. And a single neuron in the motor cortex can synapse onto 100 plus neurons in the spinal cord, and then out of the spinal cord, we could fire up 1,000 plus skeletal muscle fibers for this single neuron. It's important that we notice how much smaller our heads are than our bodies. We're going to have this concentrated effect from our heads, fewer cells within doing a lot more at the end result of a desired action or motion. Or going in an ascending fashion, we can go ahead and have a similar recruitment ascending to the CNS as well. Converging circuits then would be a concept of intensity, and within this then we have multiple inputs that converge on one source, and then we also have another type of convergence that is from one source. Do we have convergence from a single source or from multiple sources? And in any case, we have an outcome that creates greater intensity from these circuits. So these can be both sensory and motor. They have a concentrating effect, and examples of this could be different sensory stimuli leading to the same response. Reverberating and parallel after discharge circuits are important circuits for typically autonomic responses that we don't have to think about. Within these circuits then, we're interested in positive feedback, so we're going to enhance the original stimulus, and we have impulses repeatedly sent through the circuit. We see these a lot in rhythmic activities, our sleep-wake cycle, and breathing. These are circuits that thankfully last a lifetime, unless we have a problem. The parallel after discharge circuits then are many neurons arranged in parallel that stimulate a single neuron for output. This can lead to a burst of action potentials in the single neuron after discharge that can last 15 plus milliseconds after the initial input ends. What this is important for would be some of our complex mental processing. Say, for instance, doing math. We're going from one input, we're pulling in multiple other sources, and then we have this output that allows us to pull from multiple neuronal input and arrive at some higher end answers. Common neurotransmitters. We've looked a lot at acetylcholine. We also have to consider many other neurotransmitters and more that are being discovered all the time. Popular ones that we will work with and hear about are norepinephrine, dopamine, serotonin, histamine, the amino acids, GABA, glutamate, and glycine, and the peptides, the endorphins, somatostatin, and cholecystokinin. One of the things that we see within the efficiency of our system is that we can start with one element, and through a relatively simple adjustment, say the addition of oxygen, we can go from tyrosine to L-DOPA. And we also are typically, when we make these changes, we're plugging in some kind of an enzyme. Then we can kick off some CO2, use a DOPA decarboxylase, and we make dopamine. Then we can use another enzyme, and we can make norepinephrine. Then we can use another enzyme and make epinephrine. At any point along the way, if we need these neurotransmitters, they can kick off and be used within our system, or they can continue within their circulation to be adjusted by an enzyme to become a whole nother neurotransmitter. Neurotransmitter variety, then, there are over 50 potential neurotransmitters that have been discovered so far, and neurons secrete different neurotransmitters. Some neurons secrete only one, for instance, at our neuromuscular junction, acetylcholine, but most of our neurons are going to secrete two, in sequence or at once, depending on which neurotransmitters they are and where they're located. Acetylcholine is the principal neurotransmitter in the parasympathetic nervous systems. Parasympathetic is our rest and digest, and it is released at a neuromuscular junction. Biogenic amines. Amines are just certain functional groups that we go ahead and manufacture within our bodies. Norepinephrine is sympathetic nervous system component. It's a mood elevator. Epinephrine is a hormone from the adrenal gland, and we see that in the fight or flight response, so it's also within this sympathetic nervous system arena. Serotonin within the brainstem as well is inhibitory. It's key, this inhibition. It's active in mood and appetitive behavior. Amino acids. GABA aminobutyric acid in the central nervous system is the primary inhibitory neurotransmitter of the brain. So a lot of times if we see problems with ADD or issues that we need to regulate in terms of we need increased inhibition, we will most likely see GABA at play. Glutamate is the primary excitatory neurotransmitter of the brain, key for learning and memory, yet excess amounts of glutamate can cause neuron death. Oxygen deprivation and stroke is an example. Peptides are endorphins, and endorphins are elements that we have not been able to synthesize in terms of, say, for instance, if you think of a similar class of opiate, if we were to look at heroin or morphine, our endogenous or internal endorphins that we get just when we smile or have sex or eat something we enjoy are much stronger than these opiates that we could take in externally. They block pain and cause euphoria. When we think about regeneration within the central nervous system, we typically think that it doesn't happen. But this isn't entirely true. In the peripheral nervous system, if we have damage to the peripheral nervous system and our cell body is intact, so we still have our axonal cell body intact, and yet maybe we've fragmented part of the axon, say a crushing disease or maybe a surgery. If this area is close enough and if our endoneurium is available, then it is possible that this fragmented axon, for instance, can have a certain amount of healing capacity. So sometimes when people have a surgery, they might be told that they may recover some of their feeling over time, and a big part of that concept is based on this idea that in some cases, if there's enough of the axon left, then and the cell body's intact, then we may see healing occur within PNS axons. When we take a look at the cardiac action potential, it's a little different. than what we've looked at within our skeletal muscle action potential. We have a very rapid intake of sodium and a gradual output of potassium with a steep drop-off. A key thing here is we have a much greater absolute refractory period within cardiac muscle. And another player that we don't see in our action potential model for skeletal muscle is the calcium. But we still see our sodium potassium ATPase pump, and we also have a little different resting membrane potential, but a similar depolarized membrane potential. And those are some of the differences between cardiac and skeletal muscle action potential. So we're still going to have a stimulus that is going to go ahead and create similar changes of sodium in and potassium out, as well as calcium coming in as potassium's going out. These changes then go ahead and create an action potential that we still reset through ATP and our sodium potassium ATPase pump. So just kind of taking a look at this then, action potential in cardiac muscle, this idea of the contraction that we have is very much a normal curve, yet compared to that normal curve, it's interesting to just see the difference in our action potential curve that occurs based on the difference in the repetitive and consistent action potential that we require for cardiac muscle. Some diseases that we take a look at within the central nervous system would be amyotrophic lateral sclerosis, aka Lou Gehrig's disease. It's a progressive degenerative disease of the central nervous system, characterized by weakening and thinning of skeletal muscles with a loss of motor neurons in the brain and spinal cord, a form of atrophy within these areas. And it has a direct effect on voluntary motor function. Cells within the brain are neurons that transport electrical messages to other parts of the body using chemical transmitters or neurotransmitters, is what we've been looking at. But if we have damaged or lost brain tissue, we can cut off these connections. And in Alzheimer's disease, these areas that are cut off can occur from what are considered to be protein plaques. And there's also some current research being done within the idea that Alzheimer's may be a type 3 diabetes in terms of how sugar affects neurons and how the lack of ability to manage glucose can affect neurons negatively. This is a brain that we would see within Alzheimer's disease. These raised up portions of the brain are the gyri, and these depressed portions are the sulci, or you'll see pronounced sulci. These sulci are so much further apart than in a healthy brain that it's one of the only ways that a brain could be analyzed, say through CAT scan or MRI, to maybe have a sense of if Alzheimer's disease is progressing. A lot of times, a definitive analysis of Alzheimer's disease can only be achieved post-mortem. So again, just seeing this incredible atrophy, really sad progression of the disease, and as these plaques cut off connection of neurons, and also widening our interest in our information, our knowledge about a use it or lose it principle. That even if we weren't to acquire Alzheimer's, if we didn't keep our neurons strengthened for connectivity, could we see similar atrophy? This type of atrophy is also being seen within the brains of sports players and athletes who have been seriously concussed many times, and this type of atrophy is being seen to occur within their brains, almost an early onset Alzheimer's. These senile plaques are often found when microscopy is performed on the tissue, and so these plaques are the key area of interest for research right now, and just this idea of how these plaques are forming and cutting off neuron communication is a core facet of Alzheimer's research. Again, more of those plaques seen within dementia. A number of neuritic plaques, and Congo red is just a stain that's used, this red that you see. And so this is showing small peripheral cerebral arteries that might also be involved. And there also seems to be a problem with beta amyloid precursor protein, but the exact pathogenesis is unknown currently. It's interesting that the gene coding for cerebral amyloid is on chromosome 21, and persons with trisomy 21 living to age 40 invariably develop Alzheimer's disease. So this association between a genetic chromosome location for trisomy 21 or Down syndrome, that it's inevitable for those people to live until the age of 40, that they'll develop Alzheimer's disease. So this connection is something that's being looked at as well. There are also these neurofibrillary tangles that are seen within Alzheimer's. So just kind of a very tangled mass of tissue, useless, decayed neurons that at the microscopic level you can see, as you expand this up, how atrophy occurs.